Hey there, and welcome. Today, we're going to pull back the curtain on something that affects all of us medical tests. We're going to dive into the incredible science that takes a simple number on a lab report and turns it into something you can actually truly trust with your health. Let's get into it.
I mean, think about it. You've been there, right? You get a result for your cholesterol or maybe your blood sugar, and a doctor makes a really important decision based on that one number. But what's standing behind it? What gives that number its power, its authority? Well, that's exactly what we're here to unpack.
Okay, so first things first, we need to get one thing straight. There are actually two completely different questions scientists have to answer. One is, are we even measuring the right thing? Like, is cholesterol really the best marker for heart disease? That's called biomarker validation. Super interesting, but that's a whole other story. Today, we're focused on the second question, assay validation. This is all about once we've decided to measure cholesterol, how do we know our test is getting the measurement right? To earn our trust, any new medical test has to run an absolute gauntlet of challenges. Seriously, think of this list as a brutal obstacle course, a hero's journey. It's a series of critical hurdles, from accuracy to precision to how it handles real-world chaos, that it has to clear perfectly. This is our roadmap for today. So let's kick things off with the first and, most obvious hurdle of them all, accuracy. The question couldn't be simpler, but the answer is everything. Is the number the test spits out the real number? So what do scientists really mean by accuracy? It's just how close a measurement gets to the actual true value. The gap between the test's answer and the true answer, that's called bias. And here's the key takeaway. The smaller the bias, the higher the accuracy. It really is that simple. And how do they figure this out? Well, they do something called a method comparison. They basically take the new kid on the block, our new test, and put it head-to-head with the reigning champ, a trusted gold standard test. So for sample A, you can see the new test was a little high, with a bias of plus 15. For sample C, it was a bit low, minus 20. But for sample B, bam, right on the money, a bias of 0. They do this hundreds of times to get a really clear picture of the new test's accuracy. All right, hurdle number 1 is cleared. We've checked off accuracy, but hitting the bullseye once is one thing. Can you do it over and over and over again? That brings us to our next challenge, precision.
Being accurate is awesome, for sure, but it's not enough. What good is a test that's right on average if its results are all over the map every time you run it? And that brings us right to the consistency challenge. Precision is all about one word: consistency. If you take the exact same sample and measure it 10 times, how tightly clustered are those 10 results? That's precision. And it's so important because if a test isn't precise, your doctor could get one reading on Monday and a totally different one on Wednesday, leading to all sorts of confusion.
And get this, scientists are intense about this. They don't just check for precision once, they level it up. First is repeatability, kind of like easy mode, just running it over and over in one go. Then they test intermediate precision, let's call it hard mode, with different people on different days in the same lab. And the final boss level, reproducibility. Can a lab in London get the same consistent results as a lab in Singapore? And this is the classic way to show the difference. And honestly, it's perfect. Look at the top left target. The shots are all over the place. That's not accurate or precise. Top right? That's precise. See how the shots are all clustered together? But it's not accurate. They missed the bullseye. Bottom left is closer to being accurate on average, but it's not precise at all. And then bottom right. That's the dream. High accuracy and high precision. Every shot is clustered right in the center. That's the goal. 2 hurdles down. Our test is on a roll. It's accurate, it's precise, but, every machine, every instrument has its limits. So our next big challenge is to figure out exactly where those boundaries are. A test just can't measure from absolute 0 to infinity. It doesn't work that way. We have to know its limits. And the biggest question is, what's the tiniest, teeniest amount of something it can reliably see? How low can it go? This is what's known as the limit of detection, or LOD. The best analogy is trying to hear a whisper in a really loud room. The LOD is the quietest whisper that the test can confidently pick out and say, yep, I heard that, and know for sure it wasn't just part of the background noise. But hold on, because there's a really crucial distinction here. At the limit of detection, the LOD, All the test can do is say, hey, it's here. I see it. That's it. It's only when you get to a slightly higher level, the limit of quantification, or LOQ, that the test is confident enough to say, not only is it here, but here's exactly how much of it there is. And this difference is critical. For an early disease screening, maybe just detecting it is enough, but for managing something like diabetes, you absolutely need to quantify the amount of glucose. So when you put all this together, you get the test's working area, what's called the analytical measurement range. It's the sweet spot. It starts at that limit of quantification and goes all the way up to some upper limit. And inside this range, we know we can trust the number. Part of that is also making sure the test has linearity, that if the amount of stuff doubles, the test's number also doubles, nice and predictably, across that whole range. Okay, our test is looking pretty solid. It's accurate, it's precise, and we know its absolute limits. But so far, all of this has been happening in a perfect, clean, controlled laboratory. Now it's time to throw it into the chaos of the real world. Real-world samples, like our blood, they're not clean. They are a complex, messy soup of thousands of different things all sloshing around. So this final gauntlet is all about making sure the test still works perfectly in the middle of all that mess. This is also where scientists test for robustness, like, can the test handle a room being a little too warm or a bit too humid and still give the right answer? Probably the biggest challenge here is what's called analytical specificity. This is the test's ability to have perfect tunnel vision, to find the one single molecule it's looking for and completely ignore the thousands of others. Because if it can't do this, something as simple as a common over-the-counter drug could make the test light up with a false positive, and that's a recipe for disaster. And this idea of specificity actually has two crucial parts. For a test like one for HIV, you need inclusivity. HIV has lots of different strains, and the test has to be able to catch every single one of them. But for something like a TB test, you need exclusivity. There are lots of related bacteria that look kind of similar but are totally harmless. The test has to be smart enough to ignore all those lookalikes and only flag the one specific bacterium that causes TB. And we've made it. We've gone through the entire gauntlet. We've seen just how much intense, rigorous work goes into making sure just one medical test is trustworthy. But so what? Why does all this matter to you? Let's bring it all home. And there it is, the gauntlet conquered. Every single check mark on this list, from accuracy all the way down to robustness, represents another layer of certainty, another reason that you can look at that number on your lab report and believe in it. Because this is the payoff. All of that incredibly detailed science allows us to do this, to take a number, a quantitative measurement, like a cholesterol reading of 210, and confidently translate it into a meaningful category, like borderline risk. Without that science of certainty underneath, these categories would be totally useless. The number becomes a guide for action. And that really leaves us with one final big thought for the future. We've spent decades building this incredible system of trust for tests that are done in labs. But now, healthcare is moving into our homes with at-home tests and wearable devices. So the big question for all of us is, how do we make absolutely sure that this same rigorous science of certainty is built into every single one of those too? Something to think about. Thanks so much for watching.